Hi students, welcome to the last part of the Chapter 5 lecture. This is referencing the Saladin textbook. We've already learned about the various tissue types that you'll find in the human body, and with this last part of the lecture I'm going to talk briefly on cell junctions, types of membranes, as well as tissue growth and repair. Okay, cell junctions. Cell junctions are simply ways that cells are connected to one another. And generally speaking, you're going to have different types of proteins that help to anchor one cell to another or to the extracellular matrix. So these are the four common types of cell junctions that I'll talk in a little bit more detail about briefly. But we have tight junctions, desmosomes, hemidesmosomes, and then gap junctions. Most cells, with a few exceptions, such as blood cells, are going to be anchored to each other, to their matrix, or both. And these various types of cell junctions are going to provide ways in which cells communicate with each other, resist mechanical stress, and control what moves through the gaps between them. Okay, so tight junction. My pen decided to work all of a sudden. Oh, now it's not working. Okay, so tight junctions. This is a linkage between two adjacent cells by transmembrane cell adhesion proteins. And these are particularly important in the stomach and small intestines, for example. They help to seal off intercellular space, so the space between adjacent cells, and that makes it difficult or impossible for substances to pass between those cells. Now, like I said, with the stomach and small intestine, because of the highly acidic environment within the stomach due to hydrochloric acid and also various digestive juices that are being added to the GI tract, it's really important that we have some system for preventing those digestive juices from seeping between cells of the epithelial layer into the connective tissue below that. So if we didn't have tight junctions there, we could have digestive juices seeping between cells and then actually damaging or essentially digesting the connective tissue that underlies that epithelial tissue. So in epithelia, they form this area that basically completely encircles each cell near its apical pole. Remember that the apical pole is basically the side of the cell or the tissue that is furthest away from the basement membrane, excuse me. So the basement membrane is what the epithelial cells are anchored to. Below that, you have the connective tissue, and then the apical pole is basically the top of the cell, or the part of the cell that's furthest away from that basement membrane. Okay, desmosomes. These are basically like patches that help to hold cells together. They keep cells from pulling apart, so it's a really good cell junction to have when mechanical stress is an issue, for example, in our cardiac muscle cells. There is a hook-like J-shaped protein that arise from the cytoskeleton. The cytoskeleton, recall, is basically just a system of protein tubules that help provide internal structure and integrity for cells. So these J-shaped proteins will link one cell to another, and they will in turn be anchored to what's known as a plaque on the intracellular side of each cell. So these desmosomes are basically another form of transmembrane protein. So again, it's spanning the whole space within that lipid bilayer of the plasma membrane. And then we also have what's known as a hemidesmosome, which is basically half of a desmosome, and that anchors basal cells of an epithelial layer to underlying basement membrane. So if you have a simple epithelial tissue layer, that's going to hold those single cells to their basement membrane, and in the case of a stratified epithelial tissue, it's going to hold the basalmost layer of cells to that basal membrane as well, or basement membrane. And then gap junctions, these are crucial for embryonic development, for example, and also for cardiac and smooth muscle. They are ways that cells can communicate with one another, and also ways that solutes and body fluids can basically pass through these little water pores. So we have these 6 transmembrane proteins that are kind of arranged like segments of an orange around this water-filled pore, and those are called connexins. And then you have the ability for ions, which are charged particles, other nutrients, and other small solutes to directly pass from cell to cell without having to get through that phospholipid bilayer, for example. So like I said, this is important for cardiac and smooth muscle cell-to-cell communication. Also embryonic tissue before the cardiovascular system has fully formed. It's a way to distribute nutrients throughout embryonic tissue. Okay, switching gears a little bit here. Now remember that I covered types of endocrine and exocrine glands in class when I spoke about epithelial tissue. If you would like to read up more information on glands of the body, that is also included in this same general section of the Saladin textbook. But now I'm going to be speaking about membranes briefly. Membranes, of course, are oftentimes composed of epithelial tissue, but sometimes they are actually only connective tissue, or they can be a mix of many different types of tissues. So epithelial, connective, and muscular tissues, all of these can make up a membrane just depending on where this membrane is found and its specific function. So examples of membranes that are only comprised of connective tissue, the dura mater in the brain. So for example, when we get to do our sheep brain dissection in class, you'll see this very tough, sort of dry layer of material that you have to basically cut off of the brain itself, and that is going to be your protective layer around the brain that prevents direct contact between nervous tissue and the bone of our cranium. So the dura mater, both within the brain and the spinal cord, synovial membranes, which are the membranes that encapsulate joints of the body, and then periosteum, which is a connective tissue membrane that adheres to bone. And it kind of forms the medium between actual bone tissue and other tissues of the body and provides a connectivity for nutrient exchange and waste removal. So examples of membranes that are only epithelial tissue would be, for example, the anterior surfaces of the cornea and the lens of the eye. And then most of our other membranes are going to be mixtures of different types of tissues. So the peritoneum, for example, which we've talked briefly about before, the pericardium, those are going to be mixtures of multiple types of tissues. And then the skin itself can actually be classified as a membrane. It's known as the cutaneous membrane. That is the largest membrane in the body, and it's comprised, of course, of the stratified squamous epithelial tissue of our epidermis that rests on a layer of connective tissue, which is the dermis. And it's a relatively dry layer, of course, that serves protective function. Mucous membranes, also known as mucosa, are what line passages that open to the external environment, for example, our digestive tract. So oral mucosa, for example. There are several sublayers. There's the epithelium itself, which would be this layer here. There's the lamina propria, for example, areolar tissue is going to be pretty much universally found in this layer. and then the muscularis mucosa, which is a thin layer of smooth muscle. So this entire structure actually comprises a mucous membrane. And remember that epithelial tissue itself, all types of epithelial tissue, lack space for blood vessels. So the cells are very tightly packed, and as a result of that, there's no room for blood vessels to actually infiltrate that layer. So all epithelial tissue is going to rely on this underlying connective tissue for nutrient supply and waste removal. So this is a good illustration of that general pattern that we see with epithelial tissue. With mucous membranes, there are several functions. Absorptive, meaning absorbing, secretory, which means secreting things, so secreting mucus, and then protective functions as well, so protecting body passages from bacterial cells, for example, other forms of potential injury or invaders. And then often they will have mucus-producing goblet cells. And remember those goblet cells, they're kind of shaped like wine glasses. In a histological specimen, they will just appear as empty space, but in living tissue, they are going to be an exocrine, a unicellular exocrine gland that produces mucus that is then pushed over the top of the cilia in this case, and it will float on this saline layer and help to, for example, rid the upper respiratory tract of debris. We also have what are known as serous membranes, also known as serosa, and these are internal membranes, so the membranes that we learned about in our first week, the orientation to body cavities, for example, pericardium, peritoneum, and pleura. So this is a simple squamous epithelium that rests on a layer of areolar tissue. So remember that simple refers to a single layer of cells. Squamous refers to this kind of flattened scale-like shape to the cells. And underlying this is always going to be the areolar tissue. So the membrane within a membrane, remember that all epithelial tissue are adhering to this basement membrane, and then this whole structure would be considered a serous membrane or a serosa. So what do these membranes do? They produce serous fluid that arises from blood, so it's basically filtered out of blood plasma. It has a slightly different chemical composition than blood plasma itself. But this is going to be in stark contrast to the mucous membranes that are producing that thick mucin that combines with water to form mucus. A serous membrane is going to produce a much more watery fluid. And this is going to be really useful for reducing friction between organs within body cavities, for example. So you'll find serous membranes covering organs and lining the walls of body cavities. You'll also find what is known as endothelium within blood vessels and the heart. And then, of course, mesothelium lines body cavities. And these terms, endothelium and mesothelium, these ultimately derive from layers of tissue that develop in a developing fetus. Okay, let's talk briefly about tissue development. You know, we all should have a fairly solid understanding that there are many different types of tissue in the human body. The four main categories, of course, being epithelial, connective, muscle, and nervous tissue. There is a little bit of plasticity in tissue types, so tissues can change types within certain limits. Stem cells, embryonic stem cells in particular, have the most potential for actually being able to change from one cell type to another. And generally, as we grow older, we have a reduced capacity for tissue differentiation, but there are still what are known as adult stem cells that allow for differentiation to a degree. So differentiation is simply the development of more specialized form and function from an unspecialized kind of precursor tissue. The example here would be embryonic mesenchyme becoming muscle. So mesenchyme is kind of this generic basic tissue type that is unspecialized, and as the embryo develops, that mesenchyme becomes a more specialized muscle tissue. There is this phenomenon known as metaplasia, which is changing from one type of mature tissue to another, and this is not quite as common as what we see in the developing embryo, of course. A couple examples of this: before puberty, girls will have a simple cuboidal tissue of the vagina, and then post-puberty, that is going to change to a stratified squamous tissue. And then in people who smoke, before really taking up and becoming a chronic smoker, the bronchi of the lungs are comprised of pseudostratified columnar epithelium, and then given enough time, that will actually differentiate into stratified squamous epithelium. So you sometimes have, normal functionality in the case of a girl who has gone through puberty. That's going to allow for intercourse and childbearing to take place with this change in tissue type. And then in the other case, we have a loss of functionality. Pseudostratified columnar often is associated with goblet cells and mucus production, and oftentimes we'll have cilia as well, which helps to sweep debris out of the upper respiratory tract. And then if that differentiates into this stratified squamous epithelium, you might actually have some loss of that functionality. So, not only does a chronic smoker have an increased risk of lung cancer, for example, and just debris building up in their lungs due to smoking, but they also have this inherent loss of functionality due to this differentiation. Tissue growth, of course, what really comes to mind with tissue growth is the developing embryo and the relatively rapid growth that takes place in childhood. Tissue growth in the embryo and childhood is typically what is known as hyperplasia, which is growth through cell multiplication. So that's going to be the process of mitosis. But you can also have cells simply increasing in size rather than actually increasing in number. So hypertrophy, for example, enlargement of pre-existing cells. This happens with muscle growth through exercise. If you take up a weight training regimen, for example, it's not as if you are gaining muscle fibers necessarily. You are oftentimes just increasing the size of those individual muscle fibers through the demands that you're placing on that tissue. And then accumulation of body fat. So a person with more body fat than, say, average doesn't necessarily have more adipocytes, more fat cells than any other person. There's just more triglycerides stored in those adipocytes. So again, that's going to be an example of hypertrophy. And then we can also have abnormal tissue growth, neoplasia, development of a tumor, or what's known as a neoplasm. And this can either be benign, meaning it doesn't really cause health issues, and usually removal is more of an elective process, or it can be malignant in the case of cancer cells that metastasize and spread throughout the body. And a tumor, whether it's benign or malignant, will be comprised of abnormal, non-functional tissue. So you're not going to have the same inherent functionality that we get with our typical different types of tissues. Okay, let me talk a little bit about tissue repair, and that is going to wrap up the Chapter 5 material. So damaged tissues can be repaired in two ways, either through regeneration or through fibrosis. Regeneration is simply replacement of dead or damaged cells by the same type of cell as before. So you get a paper cut, for example. Your keratinized, stratified, squamous epithelial tissue is going to undergo mitosis. It's basically going to replace the skin cells that were damaged and died during that process, and you still have basically the same functionality. So you have restoration of normal function, repair of minor skin injuries, this is pretty common, and then also liver injuries as well, to a degree. The liver is pretty good at repairing itself. And then fibrosis is when you have replacement of damaged cells with scar tissue instead of the original tissue type. So the scar will physically hold organs together, but you don't have the restoration of your normal function from that pre-existing type of tissue. So if you have repair of severe cuts and burns or scarring of lungs in tuberculosis, you're actually going to see scar tissue forming there, which is fibrosis, rather than actual regeneration. And then tissue shrinkage and death. There are a number of different ways that cells will either shrink in size or number or actually die. And some of these are a result of disease or injury, and some of these are actually just a natural part of a cell's life cycle. Atrophy is loss of cell size or number. For example, if you break your leg, you can develop disuse atrophy from lack of use if your leg is in a cast. Necrosis would be pathological death of tissue, and there's a number of different examples here. Gangrene, insufficient blood supply. This is often a problem with diabetes, for example, in the extremities. Gas gangrene, which occurs when you have anaerobic bacterial infection. So if you have a deep injury, for example, that's contaminated with soil, there's actually a genus of bacterium known as Clostridium, which can get into that wound and cause this gas gangrene. Infarction is death of tissue from lack of blood. So a myocardial infarction, for example, that is the technical term for what happens with a heart attack, because you have death of cardiac muscle tissue due to lack of oxygen, lack of blood supply to that tissue. And then a decubitus ulcer is a bed sore or a pressure sore, and that can occur when a patient has prolonged direct contact with, say, a bed or a wheelchair. It often occurs in places where bones come very close to the surface of the body, so like the ankle or the hip or the sacral region, and it basically results from blood supply being cut off there. Apoptosis is programmed cell death, and this is where cells shrink and are phagocytized, which means they are basically eaten by specialized cell types, and the nutrients from the parts of the cell can basically be reused elsewhere in the body. No inflammation occurs in this case, and oftentimes this is just a natural part of the cell cycle. A good example of this is the uterus. During and immediately after pregnancy, the uterus is substantially enlarged from normal, and then several weeks after birth has occurred, programmed cell death will cause the uterus to become much smaller in size. So that is a natural process. It's not necessarily a bad thing, and this is actually a process that occurs in many of our tissues on a daily basis. Okay, as always, please let me know if you have questions about any of this material. I apologize for my pen not working, not sure what's going on there, but let me know if you have questions, and I will see you in class. Thank you for listening.